Thakur, Assistant Professor at Department of Pharmaceutical Technology, MIET, Meerut, Uttar Pradesh, heartily welcome you all on the second day of Atal Academic sponsored 5 days training program on applications of biotechnological and analytical tools in pharmaceutical research. I welcome our key speaker of first session, Dr. Abhinav Garg, who is a senior R&D professional at Arbro Pharmaceuticals Private Limited. I take an opportunity to introduce Sir. Sir is a pharma nutra testing business leader with over 10 years of strategic leadership and management experience in product and process development, testing support, continuous improvement, strategy planning, quality controls, testing compliances, regulatory and scientific affairs, drug approval, FDA audits, quality assurance, CDMO, project management, budgeting, P2P business input support, lean management, and global client support. With this, Sir has worked with number of reputed national and multinational companies and international clients for their product challenges and support in fixed timelines. Talking about his academic achievements, Sir has done his B Pharm, M Pharm, and PhD from Jamia Hamdard. Also, Sir has 2 patents in his name, which is truly inspiring. Now, without any further ado, we will turn the time over to our speaker, Dr. Abhinav Garg. Over to you, sir.
Good morning. Am I audible?
Yes, sir.
Yes, sir. You're audible.
Thank you, Vipin sir. Thank you, MIT, for inviting me, giving me an opportunity. It's a pleasure. When I saw the topic, it's really very good topic. FTIR IR spectroscopy. It's a very good topic in today's arena. I would just like to share my screen to start up. Dr. Vandana can't able to share my screen actually.
Okay, so let me check. Sir, you are a co-host. You are already a co-host. So I think you should be able to share, but let me ask my technical team. Sir, can you try again? Sir, we can see your screen.
So I'll just. So is it visible now? Are you can you able to see my complete screen, or it's only part of screen?
Sir, it's only a part of the screen, sir.
Yeah. It's full now?
Yeah. It's full.
Yes, sir.
Okay, very good, very good. So how many participants are there? Are there more? We have to start, or we shall wait?
So let us start. They are still joining.
Okay, okay.
Let us start.
So my topic for today's presentation is FDR spectroscopy as a green tool for quantitative analysis of drugs. So I'll start with the small one slide of this for IR spectroscopy, where we we are seeing NIR, MIR, and FIR. So basically, we are usually using only one part of the IR spectrum, that is MIR, which is from 400 to 4,000 centimeter inverse. But other part of the IR we are not using as on date, or or very we are using very less. It is NIR and FIR. So it has been in today's arena, today's world, NIR, FIR is been used and has been widely used for a lot of application. For I would ask with the audience also. So this is one video which I'm presenting from Bruker, our instrumental expert, which is basically giving some slide of some brief info about the FTI applications. I'm starting up.
हेलो
हेलो
अभिनव सर, आवाज नहीं आ रही है.
Sir, it is a presentation about basically FTI application
It is muted.
So is it now? It's coming, sir.
Sir, screen is visible, but voice is not there.
Okay, so that is basically a video which I'm showcasing about the FTR application. So basically, I want to ask with the audience also, have you got have they got the video? What are the basically I'm trying to showcase? So is anybody which have followed the video? So anybody in the audience who's like to answer for the video, what he or she has understand from the video?
Any participants?
Shall I repeat the video again?
Yes, sir. Once more.
Okay. So here we are showing the raw material, the products which we are going to test on FTI. It's a liquid sample, it's a powder sample, it's a tablet. So we are showing it's a raw material. So we are showing the products which we can readily use in FTI, which we are using as on date also in industry and in academics. So this is what they are showcasing. So it is a background check to detect the noise level, and they are doing the background check at the first hand. Hmm. Mr. Parul Sharma has something to ask. I have allowed you, Parul Sharma. So it is they are showing that we don't have to basically form a KBR pellet. We don't have to basically make the sample out of it. We just have to basically put the sample in the disk, in the lens which they have shown, and then we're going to press it, and then basically it's the assignment it will have the spectra. It's done. Further, the system has automatically told that the sample is glucose monohydrate. So it is no longer we have to basically see the peaks out of it. We have to see the basically bands, which bands are coming, which type of absorbance is there. Then we have to assess it out that it is basically this type of compound. Now there are multiple libraries available in the program. The use of artificial intelligence, which which gives you the clarity of the probable compound which you are working on, which type of compound it could be, and the percentage of intensity of the peaks which are matching, so they can fairly give you the qualitative and quantitative estimation of the product. So they have shown that how they have matched the peaks, and it is glucose monohydride. And they are using the crystal, which is basically diamond ATR. So it is a part of system where they are showing that how many admins and modules are there, and we can able to have record the basically every sample data, and there is no modification of the data as well. It's complete backtrail at that level. So it is the and that is Bruker, and but this is Perkin Elmer. Perkin Elmer is also one of the primary most company which is having their IR instruments. So they say that IR for everybody, everywhere, every day. So they say that IR can be used for every problem as on date. We have number of multi application. We have we can use for industry in wide industries like lubricants, biofuel, environmental industry, polymers, pharma, which we are working, education, nutraceuticals, herbal products. So it is matter of time that FTIR could IR would be basically universally accepted. And in the presentation you will going to see how basically IR application has been used, has been thoroughly accepted by the regulatory agencies also at the highest level, FDA. And they have been promoting actively to use it for the analytical purposes. So these are the various models which are basically working. This is basically their Spectrum 3 MIR, NIR, FIR systems. It is of greatest demand. They have Spectrum 10 or touch software where you can basically see the type of compound which is there. They have the libraries. And the beauty of IR is it is far, as we if we compare with other instruments on industry for analysis purposes. So it is quite cheaper also. It is handy also. You can basically you can make it portable also as on date. So it is much easier to use also, and you can have the real time analysis also. So IR application. So we can do fast reaction monitoring, basically rate of reaction, the process, how the reaction process is going on, identification, quantification, we all know. We can also do polymorphism. Where we can see what type of polymorphism. Usually we have polymorphism go on. We use XRD and other instruments for polymorphism to see the type of crystal or polymorph is there. Then functional group estimation for bioproducts, for basically chemistry people which are basically playing on functional groups and to have their organic compounds. So this we know. Coated material analysis, basically when we do type of coating in the materials, the tablet coating or capsule coating, there can be numerous type of coatings also in packaging material. When we use in packaging of pharmaceuticals, we have different liners of packaging material of there which have different WTR, OTR values of there, and based on the WTR and OTR values, we are basically assessing that how the basically it will perform. The product is there, product will behave in the stability part. So nanomaterial characterization, it is the academics and every researchers are working on nanomaterials as on date. So layered films, I've already told you the basically packaging layered films. So optic analysis, there's one more application which has come up, which is basically microscopy. So we can now do IR. We have tools where we can do microscopy by IR application. And usually we consider IR for only organic compounds. Now we can basically do organometallic or even metallic compounds also. So this is basically application for pharma manufacturing identification purpose. So we usually has a setup where everything is based on quality. Everything we are doing, we are working for quality, and it is basically through strenuous quality checks we are basically manufacturing products. So a lot of time and energy and basically system is basically built to have a quality oriented system. So we usually tend to waste a lot of time, also a lot of energy, also a lot of expenditure also on the basic daily routine. So FDA requires pharma companies to verify the identification for 100% of their incoming active ingredients, which includes assay also, plus 33% of excipients. So we can very well show that once the raw material is been procured, we have to assess its quality. We have to basically identify it whether it is of same type of node and whether it complies with the standard assay norms or not, plus excipients. So we are basically putting, so here this photo is basically showcasing how basically IR or FTNIR or near infrared system has been used to basically, they are basically probing it out, probing the substance in the real time manner and basically seeing the quality of the product. So the beauty of FTNR system in the real time analysis, that we don't have to basically prepare the sample, we don't have to even take the sample, we just have to directly just have to do the right sample analysis. So it is very easy when we don't have to apply it, we don't have to basically stop the process also. At that time, the person which is basically doing the quality check can do the real time quality analysis without any stoppage in the process. So you can imagine that what is the type of productivity, what is the type of system it would have been. Yes, Dr. Shiva Duraj, please have your comment.
Dr.
Hello,
Dr. Shiva
Shiva.
Duraj?
Yes, sir. I have unmuted, sir. Thank you.
Okay.
Yes, sir. Please go ahead.
Sir, actually, it was by mistake from his side. We can continue.
Okay. So analysis through pouches, even the layered system, where the layered RM material or finished product, we can basically do those that analysis. So transportation in quarantine, there is quarantine time where it is at the transportation stage, where there also we can do the quick NIR testing. So glass, vials, petri dish, even the granule samples, the type of granules, the type of basically the type of flow properties which they are having, bigger granules, smaller granule, uniform granule, homogeneous, heterogeneous granules, all type of those things, whether we need more mixing or not, homogeneity, all type of these things we can assess in real time manner. So you can imagine how much amount of energy, solvents, precious analytical time we can able to save by using an NIR system or FTR systems in pharma manufacturing. So now advancements in IR. So like I've said that we are using IR microscopy also, which is as on date in very in use, where we are basically imagining compound by IR microscopy. So the other thing is TGA-IR hyphenation, where we are used thermogravity analysis with IR in combination. Basically, two things hand in hand. Even we are using GC-IR, where GC and IR has been using. We have seen GC. We never seen GC with IR combination, but now GC-IR is also is been processed. So this is the analysis which has been showing that how basically modern NIR spectrometers are capable of achieving precision in range of 0.004 to 0.5% when measuring OH, NH, and CH groups of unknown compound. So the lady has been showing the photograph has been real time taking is making analysis of the compound in a pouch and the other two photos has been basically the loop where they're testing the liquid sample, and one in which we are putting the cannula into the powder, and we are testing that for the routine measurement of those products. You can see how we can basically explore it out for the IR for our routine analysis and even for the R&D side. Other application is basically NIR for dispersibility, or for basically to see the type of particle which we are working. So we are basically here using the diffraction analysis, where we can see the particle shape which we are putting in the system, and we can see that what type of diffraction analysis is there, and thereby we can form an analysis that whether the particle which we are working is basically homogeneous or basically have a heterogeneous type of setup. So unknown sample, in this I want to basically focus on that basically how we are working as on date. So in today's type of machine, we have basically set of libraries, set of programs where we are using artificial intelligence, where we can directly correlate the type of product which we are basically which we have analyzed. We need not to basically make it a estimation of the compound, or with the analysis, the prognosis of the compound is so good that the machine will going to tell you the type of product you are basically working, or what type of product it is. So the libraries, we just have to basically purchase those libraries, and they're and quite cheap libraries also. So the researcher has a basically a good advantage while using these libraries, even if they are not technical enough, or even they have some doubt. These libraries will going to give you fair good idea. So this is an example where the unknown sample has been masked by the adhesive value. So even in Arbro, we have made our library where we have basically done all the work, all the standards. We have made the standards library, and when the party or the client sends any product for their for the estimation or the quality checks, we will just compare with the standards. So it is like a sort of library which we have made, and we are basically building up for the other systems. So when we are working on some unknown compounds, or even if we are working for in some formulations where we are using n type of excipients, or even if we are working for reverse engineering of any products, so we can use FTIR analysis, where we can basically assess or see the intensity of the unknown compounds which are been coming, and the libraries were going to tell them what type of compound it can be, and we can basically do the probability of the or the permutation combination of those compounds which we can use on those systems. Of course, we know that if it is a polymers, so these polymers, it can be, if it is a drugs or some other things, if there is a coating, if it is a sustained release system, what type of coating is this, which because thickness do play a good role in the IR setup also. So you can very well assess which type of compound has this type, how much type of what is the thickness of that layer. So you can basically if I am a researcher, so I can have a fair understanding that how I have to basically proceed my research methodology or my plan of work. So this is an example of polymorphism, where we are using far IR region, and where we are basically assessing the sulfametazole form 1 and form 2. And we can very well see that basically in the same spectral range, the two more polymorphs have different spectra. So we can assess that using IR, we are basically even doing the polymorphism. So it is a very I have gone through lot of research papers, lot of studies where we are been using IR, and the good part is that IR is no longer is for only analytical processes or the in vitro processes. Now now even hospital setups or clinical physiologists or clinicians are basically assessing these IR setups for their disease prognosis, and for even the critical system units we are using IR. So this is the most the good thing which has happened recently in only August 21, where FDA has allowed to use NIR for the ANDAs and NDAs, basically analytical setups. So this is in which we are been using NIR analytical procedures for the in the pharmaceutical industry for identification, testing, assay, measurements perform on pharmaceutical starting materials, intermediates, finished products, or monitoring and controlling manufacturing processes. So process analytical guide techniques, guidance, and ICS-Q2R1 provide information about the analytical procedures. The guidance provide recommendations for development, validation, use of NIR-based analytical procedures in evaluating the identity, strength, quality, purity, potency of drug substances and drug products. So these recommendations do apply in NDAs and ANDAs and supplemental NDAs and ANDAs of small molecule drugs also. And this NIR development method development is also applicable to biological products. So it has just happened in August 2021. So recently, 1 month back. So it is a very good achievement I would say for the IR principles or IR spectroscopy, where usually previously IR is not being used as a qualitative is only used for the qualitative tool, but now FDA has allowed IR for the quantitative estimation also. And even not only quantitative estimation, even they have been promoting to give their supplemental data in the form of IR technology, and they have been readily promoting it. And process analytical technology is such a wonderful technique where we can basically make methods for real time estimation of the products. So even the companies, the Indian biggest players, the ACG, the biggest companies have been working in process analytical technology, or specifically NIR technologies, where we can readily assess the quality of granules, the quality of raw material, the quality of uh, tablets, and without doing any quality control checks, and simply by mere use of NIR, we can basically we need not to basically waste time in every step, and basically stop the process at there. We can real timely use the methods from the from raw material to basically granulation to basically tableting to basically packing, and we can assess the quality of the products at that stage only. So it is a very good achievement. It has happened. So industry would really welcome it to basically make those press processes. So I would like to ask or suggest my all the academician and researchers who basically start working on these methods, so that we can basically real time assess our basically products, and basically we can have those methods available, and even industry would readily want to take those methods. In industry we don't want to basically waste time, or basically we can't shed enough time for these type of products, these type of processes. So we would readily take those processes, and if they are available and they are validated and are robust, industry people would readily take those methods. So this is one example which I want to share, that how basically non-invasive real-time glucose monitoring has been happening by NIR technology. So the machine is a BioPi, where it has a probe, and they apply the probe in basically ear lobe, and through NIR, and it has been it is applied through Android. It is basically connected with the Android app. We can readily have the glucose estimation, and we can basically check real-time estimation of glucose without piercing or taking blood samples and do the analysis. We can readily do the glucose estimates in real-time manner. It is completely non-invasive. You can imagine the people having the type 1 diabetes, all the elderlies or senior citizens, which are basically very low on muscles, and they have to prick every day or every hour for the blood glucose level, and how blood glucose level has been basically diabetes is the basically biggest chronic disease we are in. So how NIR has been basically working in very good way, and and they have developed the system, and they have been working on it to further modify it, improve it, sorry. So this is the very good study which I want to showcase over here, and it is very new study. It is only 2021. It is two articles, one is published in the Lancet journal, and other is in elsewhere, atherosclerosis, and they are all latest articles. So I have gone through where we have seen that clinician has devised a method where they can able to see the plaques in the body also, in the plaques in the body in the by using near infrared system, and basically they can able to real time detect that the type of plaques they are having, and have those system validated also, and they have basically used, and they have basically and it's a very good means of using NIR, and in the biological physiological setups. So it is a recent article, identification of vulnerable plaques and patients by intracoronary near infrared spectroscopy and ultrasound as prospective in natural, and it has been published in March 2021. And other is a lipid-rich plaques detected in near infrared spectroscopy to predict the coronary events irrespective of any age. So other is the basically use of NIR in the hospital setup, in the intensive care setup, where we can see a patient which is basically which is having hemorrhage or stroke, and it's a bedridden patient, and in the intensive care unit they have applied the probes on the forehead of the person, two probes, and this red part is seeing the oxygen saturation level in the brain, and basically that oxygen saturation level is automatically in real time is assessed by the by the ICU or the pulse system, and it's been automatic system. There is no actual manual or personal human involvement involved, and it's been processed like that it can basically assess the brain oxygen level, and we can somehow make systems so that we can increase the brain oxygen, and the process of recovery can be we know that in the stroke or in any hemorrhage, the time is the biggest factor. As we can increase the time, we just lower the time, and we can increase the recovery, or we increase the oxygen saturation level in the brain. So the percentage of necrosis or brain cell dying can be significantly reduced. So you can very well see that it is also a 2021 article, and it is basically using detection of delayed cerebral ischemia. So ischemia usually happen when we have a poor oxygen flow. So you can see that even doctors has been basically playing on the technical side of IRs and how they are basically using it. So other very good application for the same brain purpose is where exercise effects on brain and muscle function in acutely hospitalized or older patient has been used. So it is also a 2021 article where basically real time or preventive methods can become a path breaking technologies for senior citizens who can measure brain oxygen level just as pulse oximeter for the oxygen saturation in blood. So for older people where we don't know what is the saturation level of oxygen in their brain and what type of basically prognosis they could have been in some time, and it can lead to any fatal disease or fatal happening, so we can prevent it. So we can basically work on the methods where we can do real time analysis, and we can basically prevent any morbidity also associated with it. So this is the another food application where we can see that how we can measure moisture, soluble solids content, water content, stiffness, or even even the presence of internal damage in a sealed fruit. So in IR and IR we can able to assess it. So this is the infrared microscopy for the packaging material. So every packaging layer has a different absorbance, and if it is a layered type of laminate, so we can assess through ATR IR microscopy. So this is the spectrum 2 FTR of Perkin-Elmer, where they have shown that the polyethylene, polypropylene, polystyrene, PTFE has different wavelengths. So I have seen that basically the type of packing material which you are using do play a very good role in basically product packaging, and sometimes even we can basically reduce cost as well if we know what type of packaging we have to use in the product, because sometimes packaging do play a very bigger role in the product cost, and we can we can significantly reduce basically the product cost by increasing by seeing the type of packaging which we have to use. So this is the type of graph where we have used the number of different polymer percentages, that is based on PP and PE, 12% PE, 12% PP, 9%, 6% PP, all are having different type of graph. So we can very well use for the different films and different liners. So this is the application part which I have basically covered in the first part. Now if anyone has to ask anything onto it, so we can basically bring their questions into it. Then otherwise we can start with the Atr Spectroscopy Basics.
Dear participants, sir is open for Q&A. So please write in the chat box if you have any questions. If not, you can simply write no so that we can proceed further. Okay, sir, we have received one question from Amardeep. Quantification calculation in IR. Sorry, Amardeep, I think the question is not clear. Could you elaborate what you are asking?
So I basically got the point of Amardeep.
Okay.
Basically, he is asking about the quantification, how we can use it for the quantification part. So basically, Amardeep, previously the machines which we are using, the algorithm which they have, the basically interference or noise levels which they have is not is accurate. The system process is not up to mark. But now basically the techniques or the probes or the basically disk which we are using is basically very sophisticated, and now we can basically reduce the basically noise level, the interference level, which is basically the biggest factor which we are basically which we could due to which we could not able to assess IR for the quantitative estimation. But in today's scenario, we have basically ATR and IR coming up, which we have to basically devise a method. We have to calibrate it out, just like we do in UV or HPLC. We have to make a number of runs. We have to change the systems. We have to change the machines or the person or places. as we do in the validation for the analytical processes. So by going those through rigorous steps, then we can basically make it make a process which basically is fairly quantifiable. And by making a calibration curve, then you can basically can make the calculation also in IR for the quantification. And for the identification, I have told you that I would told everyone that basically we have basically libraries coming up in the IR, the algorithms or the compounds they have. They can basically give you the fair idea of the intensity of the compound which it can have. So for example, I'll give you the example. If we are working in cosmetics, so we know that there are four or five ingredients which are basically being used as a base. Or somebody makes some huge claims for basically it is basically preservative free, or it is basically doesn't have any inorganic excipients. So we can basically put a drop of that cosmetic in a probe of IR, and basically we can see the type of compounds which they are coming up. So we have gone through one study where we have to do cosmetic reverse engineering. So we have seen that they have used only glycols in making the cosmetic, and due to fairly by using glycols, they are claiming they're claiming it doesn't have any preservatives, because glycols, we know that basically it has it has osmotic activity, and by osmotic activity it can basically reduce the type of preservatives which we can able to play on. I hope to the Amardeep, your question I could able to answer your question.
Thank you, sir.
So any other question if you have?
Yes. So we have received one more question from Kamal. Yeah. Why need to convert time domain spectra into frequency domain?
Can you come up again?
Yeah, sure. Why there is a need to convert time domain spectra into frequency domain?
So I need to basically say that time domain, or basically we know that there is various principles which we are working, basically time domain or frequency. So in the coming presentation, you can easily have the answer that why we are doing it, why there is multiple wavelengths we are working on, what type of reflectance or principle we are going to have. So basics that in the basics it would going to cover it up.
Sir, there's one more question. Is there any software available for the interpretation of FTIR results? The question is from Dr. Shiva.
So Dr. Shiva, I have already told you that basically the companies from where you have purchased your IR system, they will be providing you the software libraries. For example, I'll give you the example. I'll give you the code of a machine. The Perkin Elmel machine for IR is basically about 14 to 15 lakhs. And their software for running for they have a library for health supplements. They have a library for organic chemistry. They have a library for food products or for pharma products. So you can basically select your field of research which you are working on, and you can ask for a particular library which you want to have. So and a particular library would charge about 2 lakh rupees or two and a half for that, and it can give you complete set of compounds which they have in the library, and you can basically you can basically research with your products. So it is quite handy also, and it is, if you compare with other instrumental techniques like NMR, MAS, HPLC also, the type of easiness or portability it gives as on date is far ahead. And considering that FDAs have also allowed the system, you can very really vouch that what type of advancement it has done through time.
Any other questions? Sir, I think we can start with the IR spectroscopy basics.
So we are just starting with the IR spectroscopy basics. So I know that audience is basically faculty. So I didn't make very much of basics, but I have just tried to cover up what are the frequency or what are the basically ranges which we are working in. So we know that the infrared is basically between 2.5 micrometer to 15 micrometer. Beyond that, it is visible. Beyond that, it is X-ray and basically microwave and radio. So we are usually working in mid IR region, but now basically with advancement we are now working in NIR region and FIR region also. So the region near visible is NIR, and region near microwave is FIR. So infrared radiation lies between visible and microwave. Infrared waves have longer wavelengths than visible and shorter than microwaves. The infrared region is divided into near, mid, and far infrared. The near infrared refers to a part of infrared spectrum that is closest to visible light, and far infrared refers to a part that is closest to microwave region. The mid infrared region is between the two. For chemical analysis, we know that we are working in the mid IR region between 2.5 micrometer to 15 micrometer. So we usually use this unit wave number in IR, that is inverse. So infrared spectroscopy. So when IR light goes through a sample, some IR energy is absorbed and some is transmitted. So every compound has its unique absorbance and its transmittance, and based on those uniqueness and the type of groups which they are associated, they tend to stretch or bend, and they tend to have different type of spectra. So it says that no two unique molecular structure produce the same infrared spectrum, which makes it a fingerprint type of method, where we can have a fingerprinting of a molecule. This is due to the same fact we can able to make it in a biological process system also, because we know that in the biological process also, even in edema or in ischemia or in any other processes, we know that there is a change in the bodily fluids. If it is edema, we know that there is necrosis going on, or if it is a there is a lot of fluids out of it, so there is change in oxygen levels also, there is change in organic chemistry of the compounds also at that stage. So we can devise a method, we are devising the method. So you can imagine that if you can able to basically use the systems for your in vivo activity using NIR, you can fairly give the idea of the chemistry which is happening in those levels. Now you can basically simulate the bio activity with the chemical tools also, with the analytical tools also. So that the beauty of data which will going to come is much good, because saying that it has reduced inflammation, but saying with the qualitative and quantitative estimation by IR spectroscopy, that we have got the backing with the IR spectroscopy, it's much worthwhile, and it's confirmatory. So I would ask all my researchers that basically they should start into these methods to make these tools as widely usable, and we can basically bring a lot of value into these systems. So this is two unique molecular structure to the same infrared spectrum. So we know there are two type of vibrations: stretching vibrations and bending vibrations. So stretching, when it is stretching is also of two types: symmetric stretching, asymmetric stretching. And bending is basically scissoring, rocking, twisting, and wagging. Uh, asymmetric stretching is more than symmetric stretching, and symmetric stretching is more than basically bending vibrations. IR active species. So molecule with small energy differences between various vibrational and rotational states. Only bonds which have significant dipole moments will absorb infrared radiation. Bonds which do not absorb infrared molecule includes symmetrically substituted alkenes and alkynes, symmetric diacritic molecules like hydrogen, chloride, oxygen, nitrogen. Ionic salts like sodium chloride, KBr absorb only in far IR regions. So we can basically suitably use as a sample holder for those regions. Fingerprint regions of IR spectrum, functional region, fingerprint region. So 2000 to 500 wave number is basically is called at the fingerprint region, and 4000 to 2000 is basically called as functional region compound, where we have the stretching also and bending also. So based on these values, we know that that carbonyl or alkene or carbon C and double bond is basically having the values between 2000 to 1500, and similarly CO, C and CC is have the wave number of 1000. This is the all brush up we all know. So this is the instrumentation part where infrared source is basically is been transmitted through sample. There is a certain part which basically absorb and certain is transmitted. So we use the transmittance. And once it is there is no transmittance over here. There is high transmittance over here. So similarly over here there is no transmittance, is high transmittation, and these are the basically waves at the wavelength. So ideal spectrometer instrument has illumination source, wavelength dispersion, sample compartment, single signal detector, and computer for basically controlling data acquisition. So illumination source is a light source which provide electromagnetic energy source. Wavelength dispersion basically which disperses the interference device for separating a frequency from each other. Sample compartment which holds the sample. And signal detector basically it is the basically piezoelectric source which is basically which see the temperature change in temperature and converts into the signal, and computer it basically acquire the data. So type of sources: infrared radiation can be produced electrically by heating a source, often nurse filament or global, at 10,000 to 1800 Celsius. Nurse filament is fabricated from oxides of zirconium, thorium, cerium, and global is a small rod of silicon carbide. Detectors. So there are two types of detectors which are being used: thermal detectors, piezoelectric detectors. So thermal detectors are more or less obsolete now. They have a lot of thermal noise. So it creates an interference problem. So basically now we are basically using piezoelectric detectors, and they are being used for FTR systems. So how they work basically. IR radiation reaches the detector, and there is a temperature change producing the current that is proportional to the rate of change in temperature, and we exhibit FTR response. Scanning is types scanning the instruments and multiplex instruments. So dispersion spectrometer is an older technique, and multiplex instrument is the FTR spectrometer, which is now we are used. So how dispersion is basically happening? IR source, a sample is there is absorbance, and that light is basically put on the mirror, and then there is this dispersive grating, and then we see through the exit slit, and via detector we use basically assess the data. So scanning instrument using a grating to resolve the radiation into different individual frequencies, and exit slit isolate the specific frequency for passage of detector. The IR spectrum is basically obtained by scanning the grating over a given wave number region after passing through the sample. So this is dispersive spectrometer. Slow scanning process measures only one frequency at a time slow acquisition. limited energy throughput, optical dispersion result in light intensity loss. So basically, they are not being used as on date. They are they are being basically obsolete now. So what is the noise level? So the number of scanning which you are doing, and each scanning there is a certain noise level. So we have to basically reduce the number of scans so that the SN ratio is also reduced. Fourier transform infrared spectrometer is basically is developed to basically increase basically to basically is a limitation to encounter the dispersive instrument mainly due to slow scanning process. So it is based on the Michelson interferometer. It produces a unique type of signal which has all the infrared frequencies encoded into it, and those infrared frequencies are not superimposable, and signal can be measured very quickly and usually in the order of 1 second. Optics. So we have a light source. We have a one movable mirror and one is a fixed mirror, and that movable and fixed mirror gives you the multiple frequencies which are not superimposable, and then it has been put in the sample chamber, and then we are basically seeing from the detector. So this is the path flow where how the light moves. So it moves from the light source, then it is being reflected, and it is moved to the movable mirror, and it's again put to the beam splitter, and then again it's go to the sample chamber, and then detector. So this this is a type of source which we are having, basically a light source which is having a one type of wave number, and dichromic light which have a different type of wave number, but they are basically superimposable. But in continuous spectrum light, third part where we have only one, we have different type of wave number, but they are not superimposable. So in Michelson spectrometer, interferometer employs the beam splitter, which takes the incoming infrared beam and divides into two optical beams. So one beam reflects from the flat mirror, which is fixed in place. The other beam reflects on a flat mirror, which is on the mechanism which allows the mirror to move at a very short distance, few millimeters. Because of one beam travels in fixed length and the other is constantly changing in the mirror moves, the signal which exists from interference is as a result of two beams interfering with each other. The resultant signal is called an interferogram, and which has a unique property that every data point which makes up the signal has information about every infrared frequency which comes from the source. So infograms via Fourier transform calculations give the spectra. So time axis time domain is being decoded by FFT, and it's give you a wave number. So FFT calculation is done by CPU and is basically converted into percentage transmitted into wave number. Background spectra. So we all know that background spectra is basically playing a crucial role in the IR. So basically the oxygen in the sample chamber, or atmospheric oxygen, or the atmospheric water which is there. So we have to do the background spectrum. So must be collected for all iron measurement. This can be compared with the measurement of the sample in the beam to determine the percentage transmittance. A single background measurement can be used for many simple sample measurements because this spectra is characteristic of the instrumentation and environment. A strong background absorption from water and carbon dioxide is in the atmosphere can be reduced by purging, and an inert gas or a dry carbon dioxide can be used as scrubbed air. So it is a sample Fourier transform, background transform, and a background transfer is been basically sample transform, and we see the transmission spectrum. So Speed, because of all the frequencies are measured simultaneously, it has speed. Sensitivity, it has dramatically improved the FTIR. Right? Detectors are much more sensitive. Optical throughput is much higher signal to noise ratio. Mechanical simplicity, the moving mirror in the interferometer is the only continuous moving part in the instrument, thus low possibility of mechanical breakdown, and it is internally calibrated as well. So the instrument has a helium neon laser as the internal wavelength calibration standard. So these instruments are self-calibrating and never need to be calibrated by the user. So we can very well see that the calibration has been already done by the company at company level. So these instruments are self-calibrating. So we need not to do basically calibrate by the external agencies also. So analysis qualitative. So through the interpretation of functional group region 4,000 to 300 centimeter inverse, the fingerprint region 1300 to 910 centimeter inverse, and aromatic region 910 to 650 centimeter. So this is a structural elucidation. And compound identification, we can refer to the reference IR spectra that matches the unknown compound. Quantitative. So based on the Beers-Lambert's law, we are also using the NIR spectrum, where the intensity of the absorption band is linear proportional to the concentration of the analyte. So quantification parameters include peak height, peak area. Integration of the band area should be done carefully to ensure maximum accuracy. Near IR region is betterly suited for quantification. So for the quantification tool, we have to see that near IR region is betterly suited. So we have to we have to see the frequencies which are in the near IR region. We have to obsolete the basically mid IR and 5 IR region for quantification. So the person is asking the name of the software. So we know that the Spectrum 10 touch software is one software which have a complete FTIR product line. So this system which is have a spotlight microscopy and imaging for frontier and spectrum, and we can use MIR, NIR, FIR regions also. And I've already told you that it can give you a mobile use, robust and routine estimation for QA and QC analysis, and it can be used for other industries also. So the performance is basically the highest stage at this level. So the systems are so stable and sturdy that we can basically see that we can even run the sample in our bare hands and in our car trunk. So we can have a portable systems, and we need not to basically calibrate on each and every level every time that we are basically change the location of a system. Usually in NMRs and HPLCs, we have to give significant time for the calibration part. But here we do need not to have any calibration done. Even we have to change the place, or we are on the move. So what are the IR disk available? So usually we use KBRS, but now there's various other tools also, and which have a different cutoff. Like silicon dioxide has a 2300 cutoff, and calcium fluoride or barium fluoride has 700 or 1150 fluoride. So we can basically we can choose the material also for which we are working on, and we can fairly have the calibration done for our system validation, for our process, for our sample calibration. And based on these materials, we can basically have some good data also, that which material and for which compound we have to work on, which have a good synergy and which cannot go, based on the peak estimation. So what are the various underlying principles now IR spectroscopy is now working with? So there are 4 type of basically principles which are for the physical properties we are working with. One is transmission, one is specular reflectance, and diffuse reflectance and attenuated total reflectance, ATR. So in transmission, we know that radiation passes through the body and is absorbed by a sample, and the remainder travels to the detector. It's transmission. And other is specular reflectance, where light reflectance, the light beams come and in shiny surface, it hits the surface, and it is equal and equal reflectance. So example of these compounds is basically films or substrates. And for transmission, the example types are basically solid, liquid, gas. Diffuse reflectance is basically where diffuse scattered light is collected when a beam of light reflects a rough matte surface or powder fiber. So where we are basically doing the granular analysis, so it is been estimated by a diffuse reflectance. The sample types are powder, fiber, solids, or with rough surface. And ATR is a technique where we require little or no sample preparation. We just in the first video where I've shown you that basically we have to just put the sample on the probe, and it will give you the result. The beam of infrared light passes through the crystal and it is reflected back, and through the process it's been collected. So again, the sample types can be solid, liquid, powder. So it can give you this quantitative estimation also. So based on these, we can suit our applications which we are basically working on, and we can betterly have those systems. So these are the basic techniques where we see that how the ATR, transmission, diffuse reflectance, and transmission thick has different type of spectra coming up. So ATR has the highest transmission, and thin and thick has different transmission line. So based on the layer thickness of the sample we are working on, the transmission the percent of transmission has different type of absorbance. So the transmission thick has different type of absorbance, and transmission thin has different type of absorption. And diffuse reflectance, where there is partial reflectance or there is superimposability, so it has different type of transmittance. So transmission sensitive to the sample thickness. So the key point here is this: transmission is sensitive to sample thickness, and ATR is intensity depends on the wavelength. So the ATR, the wavelength in which we are basically reflecting on, is the key point, and reflection is the effect of scattered light. So these are the modules where transmission has been working on. They are the basically probes. It's solid probe, it's liquid probe, it's basically gas probe. It is pouch which is working on. It is also a type of probe, and it is continuous process where the gas is coming and going. So type of sampling technique in transmission we have in solids, we have KBR pellet, Nijol, Mull, cast film. So liquids, we have a demountable cell where we can basically demount the cell and we can fill the liquids into it. And for gases, we have a gaseous cell chamber. And for reflectance, we have a specular reflectance, diffuse reflectance, and ATR. ATR is a universal ATR or a horizontal ATR. Transmission mode, KBR pellet. So basically, we mix the sample in the water passel, and we press through the hydraulic press, and we form a thin transparent disc, and that KBR pellet is put in the pellet holder. Nujol mold technique, where we put a drop of Nujol oil and then we mix the sample into it, and then we put in the probe. So this Nujol is a hard paraffin oil which has some absorbance, but that absorbance we have to check on for the sample. Liquid sampling, so mainly used for the quantitative analysis, requires known and repeatable optical path length, can have fixed and variable path length, capillary film between two windows. So we fill the material in the demountable cell, the liquid, and we see to it. gas cells, two different type of gas cells. One is with smaller gas cell, 5 to 10 centimeter, and the other is a bigger gas cell, which has a volume measurement of 5 meter, 10 meter, and 20 meter. So this is Atr reflectance mode, which is basically been used for the microscopy or for the granular quantification characterization. So we know that there is shiny surface or glass surface, and light is reflected at the angle, and diffuse reflectance, we have a part of reflectance, and other is superimposable lights coming up. So this is a machine where we do diffuse reflectance. So it has a 2-3 type of cup: powder cup, abrasive pad, and abrasive stick, where we can put on the sample and we can see diffuse reflectance. So Atr is basically refraction in total internal refraction. So required sample in good contact with optical element requires high refractive index of the crystal, small amount of light penetration into the sample. So this is a key thing which we do. Basically, it is high refractive index of the crystal is required. So usually products which has crystalline nature. So which are the type of Atr crystals which are available? So based on the wavelength which we have to work on, we have diamond Atr crystals as well, germanium, silicon, AMTIR, zinc, selenium. So different Atr crystals have different shortcomings and comments. So diamond, it is pressure and scratch resistant and had some higher absorption band. Otherwise, diamond has been fairly used for number of particles products. Silicon, it is hard, mechanical, and basically thermal shocks, and resistant to basic solutions. Zinc, selenium is brittle, easy to scratch with hard materials, or incompatible with acids. So we based on basically our work, our scope, we have to assess the type of crystal we have to work on. So usually industry the instrument players ask you, or basically give you the fair application, which type of basically crystals you should procure. So my presentation is basically now ended.
Thank you so much, sir, for the commendable talk and for sharing key insights on this topic. And of course, updating us with the knowledge related to recent approvals by FDA related to NIR analytical procedure, and also helping us know about the extended applications of IR in glucose estimation, coronary plaque analysis, and oxygen saturation level in the brain, that too in the real time. I must say the session was very informative. Now, despite of knowing your busy schedule, you can take a couple of questions.
20 September English language, 26 this
dear-
demonstration work will help colleges prepare their team on how to effectively conduct 3-week student induction program in your college.
Dear participants, now we are open for Q&A session. So if you have any questions, you can just drop it in the chat box. Dear participants, we are open for Q&A. So if you have any questions, please do write in the chat box. Okay, so sir, I think the basics are already known by the faculties, but again, it was truly a very informative session by you. And thank you for answering all those questions that we have received earlier. So Vipin sir, I would like to invite you.
Dr. Abhinav Garg, thank you very much for this informative session. And I think right now there is no question, because in between you have taken all the questions and you have solved all the queries. So once again, I would like to thank you. And for the participants, I would say next session, second session, we will be starting at 12. So we will have break till then, and next session we will be starting at 12. Thank you, Abhinav sir.
Thank you, sir. Thanks for the opportunity. I appreciate that all the faculty has taken much time of theirs, and I would really appreciate if MIT would go do anything into NI or FTR area, and any industry support or anything which we would like to have, we will be happy to give them.
Thank you, sir. Thank you, sir. Definitely, we will like to have if we will doing anything on FTIR or any analytical instrument, we will like to collaborate with you.
I would like, sir, that you and all the M.Pharm PhD should take the advantage of the technologies which have been there, and you can see that how we can able to correlate the pharma and clinical practices as on date, and how basically wearable portable devices are been coming, and how price effective and cheaper technologies are there for estimation of their for their usually we see at our times usually we can't able to even analyze the compound which the product we are working on, but now the time has really changed. The algorithms, the artificial intelligence which has come is really very helpful in the research area. Thank you, sir. Thank you.
Thank you, sir. Definitely we will be collaborating with you. Thank you, sir. And thanks all the participants. Next session will start at 12.